Okay, so we have adenosine triphosphate. So basically we have the adenosine group. This here represents the adenosine. So we know that adenosine is, it is a ribose nucleic acid, and it's going to be attached to those phosphate groups. And we know that ribonucleic acid basically have that at least one phosphate when it's forming that structure. So it has the adenine structure on one end of the ribose. On the other end of it, it has that phosphate tail. Now these phosphates, they're used to basically release energy. So they store within the bonds. You have all of this energy stored within those bonds. As those bonds are clipped, or you cut those bonds, usually through hydrolysis. So as those bonds are broken, energy is going to be released. All that stored energy that's in those bonds are released. You know, that very first bond, this very first one has a whole lot of energy, right? So that provides a lot of energy for things. The second one also has energy, but nowhere near as much as that very first one. So a lot of times we just get that simple clip of adenosine triphosphate to adenosine diphosphate. Okay. You guys with me so far?
Yes.
Okay.
Yes.
So this process, by breaking it down, we're able to sit there and make energy. We're able to utilize that energy. Okay. So now catabolism is the cutting of those bonds, right? Anabolism is the generation. Okay. So we're going to be generating those ATP and other energy molecules as well too. Okay. So let's talk about them. Okay. Oh, so I should also say that when we do break that bond, so basically we get this is considered an inorganic phosphate. So you'll often see it referred to in literature and stuff like that as called PI, or inorganic phosphate. Okay. And then of course you get the ADP. Okay. So that's your first reaction. And as I said, you can actually break this down to even further. So you can get this next one broken down, then you get AMP. So then that's adenosine monophosphate. Okay. Okay, so sources of ATP. So a lot of our sources of ATP are going to be coming from our major biological large or macro biomolecules. So that's going to be your protein, that's going to be your complex carbohydrates, and that's going to be your triglycerides. So your fats, your sugars, and your protein. Okay, so when we eat, that's what we take in, right? And so that's going to be our source, our initial source of ATP, right? So, but what's happening, we're not getting that pure ATP. We're going to actually have to be, we're going to be making that ATP from these different sources by processing those foods, okay? And we're going to be getting into that in a lot of detail for this chapter, okay? So if we look at it, our food is intake. Our proteins are going to be broken down to amino acids, right? Then our amino acids are going to then be used as energy production. So then we're going to get from ADP, we're going to get the generation of ATP. Now that's the same thing with sugar. We're going to have our complex carbohydrates. Those complex carbohydrates are going to be broken down to glucose. That glucose is going to be broken down to the production of energy by using that ADP molecule and making ATP. Same thing with the fats, exactly the same way. We have our triglycerides. The triglycerides are going to be broken down to fatty acids and glycerol. That glycerol is then going to be used as an energy product, and then we're going to end up making more ATP, right? So if you think about it, we're breaking things down. So we're going through metabolism to get to anabolism. Does that make sense? So we're breaking things down to kind of build things up. So break these guys down. So all of this is catabolism. And then at the end of it, we're going to be building ATP. So we're just converting that energy from one form, a chemical form, you know, to another form, another chemical form. Okay? Good, bad, ugly?
Yes, that makes sense.
Okay, so if we are starving, right? So our body is able to sit there and kind of try to sustain itself under circumstances that we're providing stress. So we haven't eaten in days, you know, we're fasting, or you know, we're trapped on an island and we can't find any food to eat. So in those situations, our body will actually catabolize that muscle tissue. It'll start eating up that muscle tissue. And so as it eats up that muscle tissue, we're going to be making that amino acids, and then we're able to feed into that system. Okay. Same thing with fat, right? So we have all of this fat storage, you know, look around my gut, you know, I have my belly, you know, especially since COVID, I got my COVID 65, you know, going on here. So I'm exaggerating, but, you know, close. So in that case, then I already had that fat stored. So then in this case, I'm going to be breaking that down in the case of starvation conditions or during weight loss. So, but the thing is, the first one that goes is going to be the muscle. Then the fat goes last. And so under those circumstances, you guys get that?
Yes.
Okay, good. I know it's Monday. Gosh, Monday, right? That's why they put those truck shows, you know, the monster trucks, Monday, or is it Sunday? I forget, you know. Sounds better on Monday. Monday Night Football. Kind of keep you exciting, you know. Look forward to, something to look forward to. Okay, so let's talk about cellular respiration. Okay, so this is the process of cellular respiration. Oh, do you guys breathe?
Yes.
Okay, so when we breathe, we're doing what?
We're taking in.
We're inhaling oxygen and releasing carbon.
Okay, so we're going to be bringing in O2. O2 is going to be coming in. Carbon dioxide is going to be going out, right? So the reason why you bring in O2 is because we're doing a process called cellular respiration. That cellular respiration is, you need that oxygen to break down sugar, right? So we call it oxidizing glucose. So we oxidize glucose through the process of glycolysis and the Krebs cycle. So oxygen is actually utilized utilized within the Krebs cycle itself. So we need O2 to come in so that we could sit there and generate CO2 coming out. Okay. Now let me go through this process. We're going to go through it in a lot of detail because this is really the nitty gritty of physiology, right? So that very first step is called glycolysis. Glycolysis is the breaking down of that 6 carbon sugar ring to basically 3 carbon sugars, right? We call that 3 carbon sugar a pyruvate, okay? That pyruvate molecule then gets changed to acetylene-CoA. So it carries that CoA factor, okay? During that process, we have a release of carbon dioxide. Okay, with me so far?
Yes.
Okay, so this very first step actually generates 2 ATPs are generated at this point. You get 2 ATP molecules generated by us just breaking down them. Okay, so that first step, glycolysis, 2 ATPs. Now, all of this here is all happening in the cytoplasm. It's happening outside of the mitochondria, okay? It only generates, again, 2 ATPs, okay? So then we get into the mitochondria, right? So this acetylene-CoA has to be changed into acetylene-CoA, otherwise it won't be transferred into the mitochondria. It won't be accepted in there. It has to go through that transition, otherwise it's not accepted, okay? It gets in here, it goes into the Krebs cycle. Krebs cycle is going to be the cycle where we're actually going to be breaking down those sugar molecules, and then we're going to be making more CO2. Now, during this process, the CO2, we're going to be utilizing that oxygen. So that oxygen is going to be utilized at this point. Okay, so the oxygen is going to be needed for that process to continue. And I'll show you it in a lot of detail in a second. we go through this Krebs cycle, we're going to be generating, again, another ATP, right? And then we're going to go through oxidation, oxidative phosphorylation. So during oxidation phosphorylation, we're going to be generating a whole bunch of ATP. At the end of the day, if I remember correctly, there's going to be 32 ATPs generated, a total amount of 32, right? Again, 2 of them are going to occur on the outside. The rest of them are going to occur on the inside, okay? And then you're also going to, there's going to be a cost of a couple of ATP. And we'll get into that here in a second. Are you with me? Okay, so cellular respiration consists of 3 stages. You have glycosylation, you have the Krebs cycle, and then you have oxidative phosphorylation. Okay, and the bulk of your phosphates are going to be coming through here, but you need this process of the Krebs cycle to kind of power this guy. Okay, because during your Krebs cycle, you're going to be generating the electrons that are going to be used to kind of make your ATP. It's going to go through a redox reaction to make your ATP. Okay. Okay. So, are you with me so far?
Yes.
Yes.
Okay. Okay, so I need to throw in here before I get too far ahead of myself. So actually, this process is all going to be regulated and partially regulated by hormones, right? So without those hormones, we need that tie-in. Without those hormones, we wouldn't go through this process. We wouldn't be making the enzymes that are going to be needed to do this process in an efficient manner. I mean, you guys all know that these reactions can occur, but typically they take forever if we don't have enzymes to speed up those reactions. And so those hormones regulate the enzymes that are going to be generated, okay? And so for catabolism, the enzymes that we're going to be using are going to be cortisol, it's going to be glucagon, and it's going to be adrenaline, okay? The ones that are going to be responsible for anabolism, or the anabolic process, those are going to be your growth hormones. They're going to be insulin-like, the insulin-like growth factor, insulin, testosterone, and estrogen. So they're all going to be responsible for the anabolic components of metabolism. Okay, so overview, right? Glycolysis. Okay, so during glycolysis, you're going to be basically using up to ATP. So we're going to start with a glucose molecule. That ATP is going to be used to sit there and break down that glucose into the C3 molecule, the pyruvate molecule, right? So it's going to basically break this guy down. Okay, so we have this first ATP. It causes this guy to be phosphorylated. So we're basically transferring a phosphate group onto our sugar, right? Our glucose molecule. That glucose, that C6 molecule, is going to then get another phosphate. So it's going to be doubly phosphorylated. And then, so you have 2 phosphates on there. Those sugars are then going to be broken down. And this sugar is going to be broken down into 2 halves, each one having phosphorylation sites. So you guys, the circles here represents that phosphorylation. This is your C3. So this is your pyruvate that's being generated, right? So this is a phosphorylated version of pyruvate. Okay, so at this point, we're going to be producing 2 NADH products, right? And so these guys NADH is going to be basically, they're like electron transporters, right? So they're going to be bringing in electrons, right? And or when it's NADH, it has the electrons. When it's just NAD plus, it doesn't have the electrons. And so it's able to sit there and capture the electrons and transport them, okay? So, so during this process, we're gonna get another phosphorylation. So we get our second phosphorylation. That molecule is gonna be then, so I'm just saying, during this process here, these electrons are gonna be utilized to help for that second phosphorylation event. And that's gonna be generated by free phosphates attaching to it in the system. Then these doubly phosphorylated free carbon sugars, they're then going to be further processed. And as a result of that, you're going to get ADP. The first phosphate is going to be added on to the ATP to make ATP. Then same thing is going to happen here. Then the second one is going to happen here, right? So you're going to lose both of your phosphates, although this guy should have already lost it. It's being lost in that first event. This is the second. So you get a total production of 4 ATP. Now, this is all in glycolysis. Remember, I told you that overall, you have 2 ATPs that are generated. Are you with me? So we used up 2 here, and we made 4. So the total number is 2 ATP. Make sense?
Yes.
Okay, good.
So while we consume, we also double the consuming?
Yeah. So isn't it pretty cool? So we're breaking down, but we double what we make. And so even though we're consuming, we're kind of reproducing what we're making, right? We're making use of that. And then not only that, we're also generating 2 NADH, which are going to be used for that event of oxidative phosphorylation, okay? And that's just going to make more ATP. So this is kind of like storing up extra ATP that we're going to be using later on, okay? Okay, so this process, the first process of the reaction is called exogonic, meaning that energy is going to be released. Energy is released in this context. We're releasing some of that energy. So that's how we're getting the production of NADH. The second part of this process is endogonic. So we're using endogonic, again, is the release of energy. Exogonic is the release of energy. Endogonic is you bringing in energy. So this part requires energy to be put in. So that requires us that phosphorylation. Phosphorylation of those free phosphates. Okay, you with me?
Yes.
Okay, major thing is part of the process is going to be exogonic, part of the process is going to be endogonic. Okay? And so you're going to have this coupling type of event going on. Okay. Okay. Aerobic versus anaerobic. I should say, when I learned it, the Krebs Cycle, it was called Krebs Cycle when I learned it. And that's what they're using in your text. But currently they call it the TCA cycle. And that stands for tricarbolic acid cycle. And the reason why it's called tricarbolic acid, tri meaning 3, carbolic. So they're basically saying 3 carbon oxygen acid cycle, okay? And that's essentially what you're going to get from that pyruvate, okay? You're going to get pyruvic acid, okay? Or it's also called the citric acid cycle, okay? Citric acid cycle is more commonly what it's called now. Okay, so this process glycosylation doesn't require oxygen. It doesn't require oxygen at all. So we're able to sit there and make those 2 ATP, there's 2 total ATP, and there's 2 NADHs, those energy forms, without oxygen. But you're only able to make 2 ATP, right? So when we look at aerobic versus anaerobic respiration, aerobic respiration requires oxygen, right? So that means you need oxygen for that. Well, the oxygen stage of respiration, cellular respiration, is after glycolysis, right? So it's at the TCA cycle, it's at the Krebs component, okay? Does that make sense?
A little bit.
Okay. So during anaerobic respiration, you're going to be converting, instead of converting glucose into pyruvate, you're going to be converting glucose into lactate, okay? So lactate is also a 3-carbon sugar. I mean, 3-carbon ring structure. And the structure's very similar. They have the same chemical likeness, but they behave differently because the structure that they have. Does that make sense?
Yes.
Okay. So, okay. So when we go through that process, because at the end of glycolysis, right, we end up with, so we end up with pyruvic acid. So at that very end of the process, without oxygen, right? If we don't have oxygen, that pyruvic component is going to end up becoming a lactic component, okay? So when it gets ready to go, it won't go into the, this is a horrible picture. It's a horrible picture in the book as well too. Okay, so let me see if I can kind of clear it up a little bit. Okay, so this here's your anaerobic. This is your aerobic. So this is the context of oxygen. So in the context of oxygen, you do that last stage, right? Where you're doing the coupling. So you have the coupling, phosphorylation with oxygen. So then in that case, you're getting your NADH, and you're going to be getting you would have already gotten your ATP, right? So you got your ATP early on. Okay. Here, you're going to get your NADH. So you're going to have an reduction event. That's going to be still produce your 2 NADPH. You're still going to produce ATP. So you're still going to get that coupling phosphorylation with oxidation. But in the context of the anaerobic component, instead of here, when you're actually making your ATP, right, your energy is going to be reduced. That's the making of your ATP molecules. And then down here, you have both cases, you're going to have pyruvate made. So you're going to end up with 2 pyruvate molecules. That pyruvate is then going to be converted to acetylene-CoA. This is in the case of O2. And this is no O2. Okay, so down here, you're still going to make those 2 pyruvate molecules, right? So, and then those pyruvate molecules are then going to be used to make lactic acid or lactate. Now, during the process of that lactic acid production, you're going to get 2 more NADH molecules. Okay, so you're still making those energies, those high energy molecules that you're going to be able to convert into ATP later on.
Okay.
But these lactate molecules are not able to get into the Krebs cycle. They're not able to be utilized, you know, and so then all you get in that case is lactate. Okay, so are you with me so far?
Yes.
Oh, I should say I made a mistake here. So here you're taking away the electrons from the NADH. So really this reaction should be going in this direction. You're taking away the electrons from the NADH, and then you're going to end up with pyruvate breaking down your NADH. Your NADH is then going to make lactate. Okay. And so at the end of the day, you're utilizing those two ATP, those two NADHs that you had used earlier to then make it. So now you're losing it. And so then it's going to go back up and then cycle in that case. Okay, so then you get pyruvate going to lactic acid plus NADH. That NADH is going to be recycled and used up here. Sit there and make more and more NADH. But you're losing that NADH that we made, where in this context, we don't lose that NADH, we keep it. Okay, then these guys are able to sit there and go into the Krebs cycle. When it's in the Krebs cycle, we sit there, you're going to make more ATP. So your ADP is going to be made into ATP. So you're going to get another ATP molecule generated here. And then you're going to get a whole bunch of them generated down here. Because remember, I told you there's like 32 total.
Mm-hmm.
So are you with me so far?
Yes.
I know it's a lot. Are you getting tired? Can't be getting tired already. Because now we get into the Krebs cycle. Now, Krebs cycle, there's a lot of things that are going to be happening. I'm going to clear the screen a little bit. Okay, so remember, the pyruvate is going to be translated to acetylchola. So during acetylchola, you're going to produce carbon dioxide. You're going to break down your pyruvic acid or your pyruvate it's going to make carbon dioxide, and you're going to be making a 2-carbon intermediate. Now this intermediate it's brief, it's only temporary. You have coenzyme A going to be added to this 2-carbon chain. Okay, so coenzyme A with the addition of coenzyme A to that 2-carbon chain, we're going to get the generation of more NADH. So we're going to produce another set of NADH here. Okay, and then we're going to get the production of acetyl-CoA. Okay, now acetyl-CoA can actually be transported into the membrane of the mitochondria. And that's what happens here. So in the membrane of the mitochondria, we have the acetyl-CoA then, and this here is acetyl-CoA. This is the structure of the acetyl-CoA. This is just our abbreviation for that two carbon chain for acetyl-CoA. Okay, so we're going to be utilizing water, right? Because we need water to sit there and if we look at the overall equation, it's C6H12O6 plus O2 yields H2O plus CO2. Okay, so it's 6 H12O6. Okay. So H12O6. So that's 6 and that's 6. Okay, so this is the chemical equation. So we know that we're going to be utilizing water, but we're also going to be gaining back water. Okay. So in this process, we're going to basically use water because water is going to be responsible for breaking things down, but it's also going to be in the context. So it's going to break one thing down so that we can sit there and generate something else. So in this case, we have the acetylene-CoA. Acetylene-CoA is going to sit there and act with the acetate, and that's going to be used to make citrate. This is the citrate molecule. So now we went from a 2-carbon chain, we added to a 4-carbon chain. Oxalate is a 4-carbon. And then that's going to make a 6-carbon chain. Okay, so then that 6-carbon is going to go through an isomerization. So it's still going to have the same chemical compound, but we're going to move things around. So in this case, we moved one of those, one of those hydrogens that were on the carbon. We're going to connect it with OH here. And then we're going to end up with isocitrate. Here, we're going to lose 1 CO2. Okay, so then we go through the process. We lose 1 carbon. So then now we have C5, right? And that's going to make the alpha keto glutarate. So alpha keto glutarate is going to be changed from alpha keto glutarate to succinyl-CoA. Succinyl-CoA is going to give us another CO2. Okay. And I should say this is 1, this is 2, this is 3. Okay. So then succinyl-CoA is going to be, it's going to use the process of going from GDP plus a phosphate to make, GDP plus the phosphate to make GTP. And then that's going to be utilized to sit there and make ATP because we're going to transfer that phosphate from GDP to ADP to make ATP. So then we get the production of an ATP. Okay, so then we go to the cycle. This process, we just kind of get rid of that CoA component, so we just get succinate. And then we produce a high energy molecule. So high energy molecule FAD. H is going to generate H2. And then this guy, we also had a high energy molecule here and here. Okay, so then we're going to go to succinate, which is a C4, to fumarate, and then fumarate is going to utilize some water, and then we're going to get malate. Okay, so then after we get the malate, we're going to sit there and transfer some electrons to make some NADH. So at this point, we have an NADH here. There's an NADH here. So now we have a total of 2 NADHs. Sorry, 3 NADHs. And 1 FADH2. Now it's called the Krebs cycle because it's a cycle. So what's going to happen is that malate is going to be used to make our oxalate acetate, and then we're going to use that to sit there and bind with the CoA as the next one goes in. So you're going to have this cycle that is going to be constantly running. Okay. So all together, we got an ATP, one ATP out of the Krebs cycle. We have 3 NADHs. We have 1 FADH2. And we have, let's see, 1, 2, that's it, 2 CO2s here. So Krebs. Okay, so before we enter the Krebs cycle, right, during glycolysis, how many ATPs, total ATPs do we have?
1.
Are you sure?
Oh, it's 2, sorry.
2, that's right. So we had 2, because it was a total of 4 that were made, but we used 2. So we have 2 ATPs. We have how many NADHs?
3.
Should be 1. And then we have at the very, before we go into here, we have the CO2 that's being generated. So we have 1 CO2. So now remember 1, 2, this is 2 CO2 here. So now we accounted for 2 CO2s. Now at the end of the day, we should end up with 6 carbon dioxides, right? Is that true?
Yes.
Okay, so let's sit there and think about that. Now, we made 2 pyruvates, right, to put in here. This only showing 1 of those. So the second one is going to generate, you're going to have 2 here all together, and then you're going to have an additional, so a total of 4 for each pyruvate. You're going to have, let me fix this then, a little differently. So we'll go 2 times, right? So we get 2 times here. We're going to get 2 times here, 2 times here, 2 times here, and 2 times here. Because this is for one molecule of pyruvate going in. And we know that we have 2 of them going in that is made from that original sugar. Are you with me? You see where I'm coming from?
Yes.
Okay, good. Okay, so we're going to keep a running tally. Okay, although I'm going to erase this part here. And then move this guy over. Put it in the black. That's an S. Okay. Oops. Okay, so now this is the ETC, or this is the stage of oxidative phosphorylation. So basically what you're doing in the case of the ETC is called the electron transport chain. And so what you're doing, you're going to be going through a gradient, right? So we're going to be actually utilizing those NADHs to sit there and make ATP. Okay, so let's see. Okay, so this here is the inner membrane of the mitochondria, and we have H plus molecules. Remember we talked about the hydrogen pumps, right? And how hydrogen pumps play a part in acid buildup and all that stuff, but they also play a part in terms of your mitochondria production as well. Okay, so inner membrane of the mitochondria, this is the mitochondrial matrix. This is the intermembrane space. So hydrogen ions are going to be transported from one side of that membrane to the other side of the membrane to generate an electron gradient. So as these guys go through, this guy is going to be kind of powered by this process, right? So we're going to utilize that NADH to sit there and allow to go from the hydrogen ion to go from one side of the membrane to the other side of the membrane. And then that's going to kind of produce that electron potential, right? So then that electron potential is going to sit there be utilized to sit there and power this entire process, right? And then as a result of that's happening, you're going to use this gradient, this H plus gradient, to sit there and make more ATP. So it's going to be a coupling of a reaction, okay? So the NADHs provide 2 electrons. As a result of 2 electrons being made, we get a total of 2 ATP. Okay, now this is where we're going to be utilizing oxygen. So those oxygen molecules are going to be utilized in this process. Oxygen is going to be split in half. So you're going to split those oxygen molecules, and then that's going to be utilized again for this process. Okay, are you with me so far?
Yes.
Okay, so then we're going to be basically generating ATP, right? So the same thing is going to be happening here with the FADH2, exactly the same thing. Okay, so it's going to be utilizing the electrons that are made, and then you're also going to be utilizing the H pluses. Okay. So if we keep that right, it shows it. Come on. I may be able to get it to move. Maybe. Okay, there we go. Okay, so glycolysis, we generated 2 ATP, right? Then we generated here 2 NADHs. Then here we had 3 NADHs times 2, so we generated a total of 6 NADHs. There. And then we generated 2 FADH. Okay, so then as we go down the gradient chain, we end up generating 3 ATPs for every 10 NADHs. So 3 for every 1 NADH, and then 2 ATPs for every 1 FADH2. So that's going to give us a total of 34. So if we add it all together, So we end up with a total of 36 ATP per glucose molecule. Are you with me?
Yes.
Okay.
Yes.
Okay, so that's basically showing us how we're actually utilizing glucose, right? So, but do we always start off with glucose? Or do we end up having to get glucose from another source?
We do end up getting it from another source.
Okay, so yes. So one of those sources, one of the processes called gluconeogenesis, right? So basically gluconeogenesis, I don't know why I'm in trouble. Okay, clear. Okay, so gluconeogenesis is basically, it stands for gluco, meaning glucose, right? Neo, meaning new, right? Genesis, synthesis. So we're making new glucose, okay? So there's a couple of ways that we can sit there and make new glucose, right? One of the sources can be from pyruvate, it can be from lactate, it can be from glycerol, it can be from adenine, it can be from glutamate, okay? So we know that, you may not know, but I know, but now you know, these guys are amino acids. This is a product of fatty acids, right? They're not fatty acids, lipids, lipid structures. Triglycerides. Exactly. Otherwise, I'll get in trouble. Okay, so we know that these guys come from triglycerides, or this guy comes from triglycerides. And then we know that these are our products from sugars, right? Okay. Good so far?
Mm-hmm.
Okay.
Yeah.
Okay, so, sorry. In the case of, so this would be the case for lactic acid or lactate. So in the case of lactate, we can start from lactate, or we can start here from pyruvate, right? This starts that whole process. So we have pyruvate, right? So then pyruvate, lactate is going to be converted to pyruvate. Pyruvate is then going to be, it's going to bind to that oxaloacetate, oxaloacetate together with that pyruvate. Now is going to it's going to be an enzyme that's going to be responsible for phosphorylation. So you're going to get phosphates added onto it. Okay, so you're going to have phosphates added onto these molecules. You can see the phosphate here. And so you're going to end up with phosphoryl enol pyruvate. So then that's going to then be generated through two of these structures, right? So we went from, again, that's 1, 2, 3, 4, 5. So from a 3 carbon to a 4 carbon, back to a 3 carbon. 3, 3, 4, 1, 2, 3. And then as a result of that, we're going to end up with 2 of those being generated. Then this phosphoenol pyruvate is then going to be made to phosphoglycerate. And then that, 2 phosphoglycerate. Then there's going to form an isomer. So then now it's going to be 3-phosphoglycerate. And then it's going to be converted to 1,3-biphosphoglycerate. So again, 2 phosphates here. You go from being on the 2 carbon, the phosphate being on the 2 carbon, to being on the 3 carbon. And then we're going to go from that 1,3-biphosphoglycerate to glyceraldehyde 3-phosphate. So in this case, we went from an acid, a carboxylic acid, to aldehyde. And then down here, we're going to go from glyceraldehyde 3-phosphate to glyceraldehyde. I'm sorry, you're going to take 2 of these glyceraldehyde 3-phosphates, and you're going to use that to make fructose 6-phosphate. Fructose is it's a 5-carbon, I should say 1, 2, 3, 4. So it's a 6-carbon, but a 5-point ring structure, a 5-ring structure. Okay, so it has 5 angles. So you have the oxygen, you have 1, 2, 3, 4, so a total of 5 points. But it's still a 6-carbon structure. Okay, so we'll then go from that fructose 6-phosphate to glucose 6-phosphate. Glucose has 6, it's a 6-ring structure. Okay, and then from that glucose 6-phosphate, it's going to be dephosphorylated. We're going to end up with just glucose. And so at the end of the day, you're going to end up with 1 glucose molecule. Okay. So, but that's in the case of lactate. But that's an example of making new sugar from that. Okay, so we're also going to get lipid metabolism. Okay, so here, this is basically a triglyceride. It's going to be broken down to a monoglyceride. Okay, so you're going to start off with 3, tri meaning 3, glyceride molecules. So here you have your glycerol backbones, right? And then these are your long carbon chains that are going to be attached here. So at the end of the day, you're going to go from this molecule, right, just to this single molecule with 1 long lipid chain. So you still have the glycerol backbone, but you only have a single carbon chain associated with it. So this process is just going to use oxidation, where you're going to be utilizing these fats, and you're just going to be breaking that oxide bonds. And so you're going to end up with a carboxylic acid and your glycerol chain. Okay. And this is all done through hydrolysis. Okay. So these are typically done or catalyzed by enzymes called lipase. And they're basically going to be breaking down these molecules. So the chylomicrons, so the chylomicrons are just, remember, they're just, they're going to be lipid structures. They're going to contain our triglycerides. They're going to contain all of our lipids in a vesicle or in a transport molecule, okay? And so if you take a look at this, so you have the phospholipids on the outside. So we know that it's membrane associated and membrane bound. And that's the reason why we're able to sit there and get these guys internalized. so easily because they're utilizing the phosphomembrane layer. You're going to have the apolipoproteins that are going to be embedded. So we also know that, again, those are lipids, those are membrane structures, okay? And then we're going to have on the inside, all of those fat. So you're going to have so when we think about this, right? On the outside of the phospholipids, those are going to be the hydrophilic heads, right? So those guys are going to be interacting with water. So these guys are able to transport and transport through the bloodstream, right? Because of the hydrophilic head. And then they're able to be absorbed because they have the lipid components as well too in the intestinal area. Okay. Are you with me so far?
Yes.
Okay. So because of the packaging of it, we're able to sit there and get those fats, those triglycerides, which are going to be very, very hydrophobic, because they're surrounded by that phospholipid component. So these guys are going to be traveling in the bloodstream. Now, the problem is that sometimes these guys are what are going to be causing the clot. So if they're too big or two things like that, they can end up getting together and clotting, causing clotting itself, because you start getting more of these components and less of these components available, and they start attaching to the membrane of the vessel, blood vessel itself. And so, these are the things that can end up causing problems with this, right? Okay. So these guys are going to be basically carried to adipose storage when they need to be stored. And then they're going to be delivered to other components when they need to be processed. Okay. So this is the breakdown of fatty acids. So here we have that fatty acid that we were talking about earlier. So that, so after we go from triglycerides to monoglycerides. So those monoglycerides, or the fatty acids that are going to be left over from those monoglycerides, are then going to be processed. So you have this here would be that fatty acid. You're going to have your fatty acids can be converted to your fatty acid acyl carnitine. Carnitine is an amino acid that is often used. It's a intermediate amino acid that is only used for fatty acid metabolism, right? And so, have you heard of people taking carnitine to help their metabolism for weight loss or muscle development? You've heard of that as a supplement.
Can you repeat that one?
Let me write it out. Have you heard of carnitine?
Oh, yes.
Okay. So carnitine is just basically, it is an amino acid that is utilized for fat metabolism. And so your fatty acid is actually going to bind to that carnitine. So you're going to have your fatty acid. It's going to first be attached to the CoA. The CoA is then going to bind it to carnitine to get fatty acetylcarnitine. Here, I know the picture's horrible. It's not that great in the book either. So then that guy is able to sit there and be trafficked to the mitochondria matrix. Once in the mitochondria matrix, the acyl fatty acid carnitine is then going to be dissociated from the carnitine and it's going to be attached to the acetyl-CoA. So then the acetyl-CoA fatty acid is then going to be converted to beta-keto acetyl-CoA. So we go from, this is our fatty acid. This guy is going to be our beta. Beta keto acetyl-CoA. Okay, so at the end of the process, that's what you're going to end up with. Okay. And so you're going to have intermediates between there. So you're going to first get your transanol, and then your transanol is going to be, it's going to be oxidized and hydrolyzed with water, causing these guys to sit there and be released. And then you're going to get your acetyl, your beta keto acetyl-CoA. And then your beta acetyl-CoA is going to be broken down to just acetyl-CoA. And one is actually going to be a shorter ketone. And then the other one is just going to be straight acetyl-CoA. And then that acetyl-CoA is then going to enter the Krebs cycle. Okay. So that's kind of how lipids enter. If you take a look at it, right, and you think about where lipid is actually entering, lipids are entering directly into, or fatty acids are entering directly into the mitochondria. So they don't have to go through that glycolysis component. Okay. Are you good so far?
Yes.
Yes.
I know it's a lot. Are your brains on fire yet?
Yes.
Okay. So then we have ketogenesis, right? Here we're going to be basically making ketones. That's the root of the matter. The ketone structure, so you know, it's going to be generically, it's going to be here, it's going to be double bond to an O, C, and then you're going to have carbon chains on both sides. That's a ketone. So this is an example of, it's almost a ketone, but you also have, since you have that CO2 at the end of it, it makes it a acetoacetate, but it has that ketone inner structure, okay? So ketogenesis is basically the formation of ketone bodies. In this process, we have two ketones, essentially. So we have the carbon and the acetyl-CoA molecules. So they're going to actually come together, and then you're going to get the aceto-acetyl-CoA. The aceto-acetyl-CoA is going to then be used to make your beta hydroxy beta methyl glutarate CoA, or your HMG CoA. You're going to lose your CoA component, and then you're just going to be left with the acetyl acetate. And then that acetyl acetate is going to go through conformational changes and the addition, or the hydrolysis, to add on your OH molecules to make your beta hydroxy butyrate. Beta hydroxy butyrate is then gonna be utilized to sit there and make some more NADH, some more energy. You're gonna get the acetyl acetate back. So then you're gonna get your CoAs delivered. So then you're gonna get acetyl acyl CoA. Acetyl acyl CoA, you're gonna end up getting 2 of those. So that you're gonna get it splitting by the addition of another CoA molecule. So you're gonna get 2 acetyl CoA. And then the 2 acetyl CoAs are gonna be able to enter the Krebs cycle. So by utilizing those fatty acids, that oxidation, so we went down here, right? And we kind of broke it up into their 2 components, the ketogenesis. Once we get the ketogenesis, the keto oxidation of the fatty acid, and then it's able to sit there and go into the Krebs cycle. Okay, so we also have lipogenesis. Lipogenesis is the, oops, oh, I got ahead of myself. I don't have lipogenesis. So there's also lipogenesis. Lipogenesis is the formation of lipid structures, right? So we can actually make lipids as well too. Okay, so and we do that basically starting from a sugar molecule, right? Okay, so if we sit there and we take a look at this, right? So we actually have, let's start. So in the mitochondria, we have the citric acid that is going to be generated, and that's going to be made from the oxoacetate acetyl-CoA. That's going to give us citric acid. Citric acid is going to be utilized to remake that oxoacetate, and then again, that acetyl-CoA. And then we're going to get oxoacetate, oxoacetic acid. Then that's going to be used to make malic acid. Malic acid is going to be used to make pyruvate. And then we know that pyruvate is going to go through that process of going to make acetyl-CoA, and then it's going to be able to enter the process again. So this acetyl-CoA then has this route where it is going to make from acetyl-CoA to malonyl CoA. That's going to be utilized to sit there and make a fatty acid. Fatty acid is going to bind to the glycerol molecule. That glycerol is going to make a monoglycerol. And then later on, we could add more fatty acids to that chain to make a triglyceride. Okay, so this process so with proteins, you know, as we eat proteins, the first thing that happens is that we actually start digesting proteins with digestive enzymes. These enzymes are going to be in the stomach. So the major enzyme is going to be pepsin. And then we also have hydrochloric acid. That's not an enzyme, it's an acid. The acid is also going to be used to sit there and break down proteins, okay? Pepsin is an enzyme. It cuts the protein into large pieces. So it'll just kind of slice up the protein. And then hydrochloric acid also is going to end up cutting the protein into large pieces. Then the pancreas, the acinar cells in the pancreas, they're going to be producing trypsin. Trypsin, chymotrypsin, elastin, and of course, sodium bicarbonate. Sodium bicarbonate is just a chemical, kind of baking soda, right? So those things are going to be released into the stomach. Okay, so trypsin then cuts even smaller. So it's going to cut the amino acids to smaller components there. So, and not in the, they're not going to be released in the stomach, and the intestinal cells, in the intestines. So these guys are going to be released in the intestines, and the intestines is going to cause the further clipping of the proteins into amino acids. And so they're going to be broken down even smaller at that point. And then you're going to have secretin and chlorocytokine. And so secretin and chlorocytokines, they're also going to be responsible for that last component to make sure that everything is good enough, and then also responsible for the collection of the amino acids so that they can actually be absorbed. Okay. This is the urea cycle, and we'll pick this up on Wednesday. I know it's a lot. How are you guys doing?
Okay.
Okay. It's not blown up yet?
No.
A little bit.
A little bit. Okay. Okay. The biggest thing about the urea cycle is this is where you're going to get a lot of your nitrogen from. And so we're going to be talking about how, you know, our body needs nitrogen. A lot of our, our amino acids and then also our nucleic acids need nitrogen, right? So when we do synthesis, de novo synthesis of those items, then we know that we're going to be getting it from this source, one of these sources, okay? Okay, I will see you guys on Wednesday then. You guys will have an exam. I'll be putting in your practice exam. This will be your last exam before your final. Your final has to be completed by the end of next Friday. And it's going to be just basically cover these last chapters that I'm going over this week. And that's it. So your exam before this, the exam that'll be posted this week, is going to cover all the chapters that we've gone up to before today. So okay. To be specific, I think it's 14, 15, 16, wait, it's 12, 14, 15, 16, 17. And then 23, 24, 25, 26 are all going to be on the final. And maybe if I get lucky, 27, 28, if I get lucky. Okay?
Okay.
Okay. Questions?
Do we also have a lab on Wednesday, or is it just?
It's just now you only have, so I wanted you guys to do one more lab with the fish, but I kind of decided, a lot of people have dead fish. I decided to let our fish live because the last one was the alcohol, you know.
Mm-hmm.
That was only the one left. And that could be, it could go very well depending on the behavior of our fish. I mean, or how our fish are surviving, or it could go very bad, you know. So I decided just to kind of table that one. And then, so all you have left are the simulations.
Okay.
Okay. And that's it.
Okay.
So just make sure you get your simulations done. You know, and upload them. I need to make sure that we have uploading spots. But that's it. Pretty simple.
Yeah.
How are you doing in chem?
I'm trying to catch up on everything.
Yeah.
I feel like it's, yeah, once you fall behind, somehow it all kind of crumbles down.
Yeah. Oh, it catches up.
But I'm getting there. I'm getting there. Should be all done before the end of the week.
Okay. Good luck. Good luck.
All righty.
Okay.
Okay. Well, see you on Wednesday. Oh, well, tomorrow, actually.
Yep. I'll see you tomorrow. Yeah,
okay.
exactly. Okay. I'll see you guys later. Take care.
All right. Bye.
Bye.
Bye-bye.